Good afternoon, everyone. We will get started with the symposium now, and the session one will be on molecular medicine. This session will have three types of lectures. One is keynote address by Dr. Gang Bao. The second will be on Aurobindo Nath Bose Entrepreneurship lecture by Dr. Venkataramanan. And the third will be lightning talks by students. So in the first keynote address by Dr. Gang Bao, the title is Nanomedicine and Genome Editing Based Approaches for Disease Therapies. And I will introduce Dr. Bao. Dr. Gang Bao is Department Chair of Foyt Family Professor of Bioengineering, Professor of Chemistry and Material Science and Nanoengineering. Dr. Bao is a pioneer in nanomedicine, molecular imaging, and the emerging area of genome editing. Bao is the author of more than 200 publications, 2 books, and more than 4 book chapters. Dr. Bao is also the co-founder of Vibonetics, which is a biotechnology company that develops gene expression visualization tools in living cells. He is member of Biomedical Engineering Society, the American Institute of Medical and Bioengineering, the American Physical Society, and many more societies. So with this short introduction, I request Dr. Bao to deliver his keynote address.
Thank you so much. So it's a pleasure to be here. So today, I will focus on the work in my lab in 2 areas. One is nanomedicine, the other is genome editing. I first would like to acknowledge funding agencies for supporting my work. Somehow the
Can we put this one? Can you put this?
No, it's not this even. This is not moving.
Can you stop this and share again?
Somehow it will not move.
Just can you stop this? Can you
Sorry, there's some technical issue.
No.
Oh, okay. Yeah, so this outline of my talk. First, I will go through the work in magnetic nanoparticle heating. Then I will show a particular application in using magnetic heating to treat cancer metastasis. Then I will show the work in my lab on treating sickle cell disease. So, as you may know, magnetic nanoparticles is a very special kind of nanoparticles. There are different crystals you can you can synthesize. Those are nanocrystals. We typically coat those nanocrystals with a biopolymer called the phospholipid PEG. The coating is biocompatible. And we could have different sizes. I will show you from 6 nanometer all the way to 40 nanometer. We can tune the coating thickness as a way to tune the size. The coating is also very stable. So in terms of application, there are 3 major areas. One is to do imaging because the magnetic nanoparticles are the contrast agent for MRI. We can also use magnetic force because if you apply a magnetic field, it generates a lot of force. The third area is really to generate heat by applying a alternating magnetic field. So as you can see here, we synthesized nanoparticles with different sizes, and those are very uniform. As you increase the size of nanoparticles, you can see the shape changes from roughly spherical to all kinds of different sizes. In fact, if the size is below 20 nanometer, it's super paramagnetic, meaning unless you apply a magnetic field, those are not magnetic. But if the size is larger than 20 nanometer, those becomes partially magnetic. So for imaging, over the years, we have done a lot of imaging work. In this particular case, we use the nanoparticle for MRI. We also put a dye. We actually could have two dyes. One is a near-infrared dye, so we can do both fluorescence imaging and MRI, showing here. So the example showing here is to use magnetic force. If we put the hot cell culture dish on top of an array of magnet as a way to apply magnetic force, we can actually drag the nanoparticles into cells cultured in the dish. So in that way, we can deliver a lot of nanoparticles in cells on the order of 50,000 per cell. So why we are doing that? Actually, one major issue in regenerative medicine is that we try to use stem cells for repairing damaged tissue. But if you inject the stem cells, they migrate away. They will not stay at the injury site. So in our cases, we use the human muscle-derived stem cells. We deliver nanoparticles into those stem cells. Showing here, you can see if we put the cells on top of a magnet with a particular shape, right? So if you do washing, you can wash away other cells, but the cells on top of this magnetic grid will stay. So that really shows the idea that we can use this as a way to keep stem cells at the injury site. We are still doing some animal studies. So for using heating, I will show you more examples. This is a particular example that is we deliver magnetic nanoparticles into the brain of flies. So the idea is that there are heat-sensitive ion channels. If you have magnetic nanoparticles next to the ion channel, when you apply an alternating magnetic field, you can use the heat to control the ion channels. You can see without applying magnetic field, the wings are in this position. Once you apply magnetic field, actually you turn on, so you can see the fly change the position of the wing. We can do that very fast. That paper was published in Nature Materials last year. So for magnetic heating, there are different mechanisms. So used to be people just believe the linear theory, which is based on the neo-relaxation and Brownian relaxation, right? So, but that's actually, I will show you, not accurate. So more recently, we believe the nonlinear theory, which is based on hysteresis, is a better theory to understand magnetic heating. So quite a few years ago, there's a very classic paper published actually in 2002. This paper has been cited more than 2,800 times. So in this theory, it predicts that the best magnetic nanoparticle size is around 14 nanometer. This is a radius, you can see that's a 7. This is a 14. If you have nanoparticles smaller than 14, the heat generation as quantified here decreases dramatically. If you have nanoparticles slightly larger than 14 nanometer, the heat generation also decreases dramatically. You can see that. So again, a lot of people in the field believe this is true, but we don't believe that. So what we did was we generate a lot of nanoparticles with different sizes. We put the nanoparticles in a tube. We put that in at the center of the coil, applying a magnetic field. So we systematically measure the heat generation at a different field strengths and showing here. So you can see this is the size of the nanoparticles. This is the heat generation measured by so-called SAR, which is specific absorption rate. That's typical in the field. You can see that as we increase the particle size, the heat generation increases, right? Plateaus at around 30 nanometer. Those are at two different field strengths. If I go back, you can see that that's very different from this sharp peak at 40 nanometer. So based on that, we have done a lot of validation. In fact, when my postdoc showed me these results, I asked him, are you sure this is true? Because this is against classic theory. So just to be sure, I sent those nanoparticles to a lab in Minnesota to do independent measurement. Actually, that confirms our results. So then we ask why, because the classic theory actually is based on linear theory. In that case, the hysteresis was neglected. That's why the theory was not really working. So we look into that. We shown that here it's indeed due to hysteresis. You can see the hysteresis loops. So if we quantify the energy loss per cycle due to hysteresis, you see this curve, which is very similar to the heat generation we measured as a function of size, right? So that means indeed it is due to hysteresis. So based on that, we develop a new model, and actually this is an analytic expression of the heat generation in terms of size. And we compared our results with showing as points at two different field strengths. The solid lines are from our model. All the other models you can see are not accurate. So this model works. So more recently, we generated another nanoparticle system called magnetic nanoclusters. You can see from this EM image that a lot of individual small nanoparticles, in fact, those are not isolated nanoparticles. They form a network. We can control the primary nanoparticle size from 4 nanometer all the way to 9 nanometer. We can control the total size of the nanoclusters from 35 nanometer to 90 nanometer. Why we are doing that? Because with smaller magnetic nanoparticles, you cannot really generate a lot of heat. But when you have larger nanoparticles, in fact, due to the shape change, the coating usually not as stable. With the nanoclusters, we could have a lot of heat generation, but we can avoid the issues with coating. So the idea of using this for cancer treatment is here. First, we use the nanoclusters for generating a lot of heat. Second, we actually conjugate a small molecule called AAPH. At temperature above 40 degrees C, the AAPH molecule decomposes into free radicals. Those are carbon-centered free radicals. We want to use both heat and free radicals to kill tumor cells. And let me show you how that works. First, we quantify the free radical generation using, you can see that only if we have nanoclusters with a small molecule AAPH, with the applied magnetic field, we saw a high level of generation of free radicals. Then we did a cellular study to show if we have the nanoclusters with AAPH, with applied magnetic field, we can kill a lot of tumor cells as shown here. So the red shows the dead cells. If we quantify that, you can see the amount of cell death is very significant if we combine heat generation and the free radical generation. So then we study the mechanism. We show that under both heating and free radical generation, the cell death, again, very significant. Not only that, we've shown that the cell death is immunogenic. Why this is important? Because we can use the cancer cell death induced by heating and free radical generation to activate the T cells. So in this case, we have a tumor model here. This is a colon cancer model. So we generated a tumor here. Then after 14 days, we injected the nanoclusters with the AAPH directly into the tumor. Then we applied the alternating magnetic field. So those are the controls with the ceiling, with just APH, with just nanoclusters, or nanoclusters with APH, but without applying magnetic field, right? So those you can see the tumor growth is pretty fast. If we have just nanoclusters without APH, the nanoclusters generate heat. So that kills tumor cells. That's why there's a slowdown. Now, if we have both heating generated by nanoclusters and APH to generate the free radicals, you can see the tumor growth dramatically reduced, right? So if we take out the tumors, we measure the size, you can see again, with heating and free radical generation, after applying alternating magnetic field, dramatic shrinkage of the tumor size. In one case, the tumor's entirely gone, right? So we quantify that. So one issue of cancer treatment is that at early stage, doctors have different ways to treat cancer, right? But at the late stage, that is after having metastasis, there's no good way to treat cancer. More than 80% of cancer patients die because of metastasis, right? So what we want to do here is to say if we can treat cancer metastasis to show that we generated two tumors, one tumor here, that's a primary tumor as before. We generate a secondary tumor. You can see the amount of cells we use to generate tumor is 1/5 of the original primary tumor, so it's a smaller tumor. So again, we inject nanoparticles into the primary tumor. We did not really touch the secondary tumor. Then we follow the growth of the secondary tumor, right? So as you can see here, those are the controls. And this is really just with magnetic heating and with AP. The third one, what we did was, in addition to the nanoclusters, we injected anti-PD-1 antibody. That is actually the immunotherapy agent, so-called the immunoblockade checkpoint therapy, right? So we combine all three. So we show that with all three together, the secondary tumor actually dramatically shrinked. The growth actually was suppressed. If we look at the survival curve, only when we have all three heat generation free radicals and with anti-PD-1 antibody, the mouse survived, right? So that means, again, we did not really touch the secondary tumor. We just inject the nanocluster into primary tumor. We show that second tumor suppressed, right? So we would like to use this further to show we can treat cancer metastasis. So one question, why? So in fact, it's due to CD8 positive T cells. The T cells were activated by the cell death in the primary tumor. So this is the amount of infiltrating T cells in the second tumor, right? There's a dramatic increase. And not only that, we also show that the T cells actually have tumor antigen specific T cells. That is, once you activate those T cells, they can recognize secondary tumors because the ability to recognize tumor antigen specific cells. So anyway, so with this, we believe there's a very good potential for this to be translated into clinical use. We are now working with MD Anderson Cancer Center to further illustrate. Now, one issue is that if you have surgery, right? If you have surgery, a lot of patients actually have tumors coming back, right? So we have shown that there's a memory effect. So in addition to the original tumor, we waited for 40 days, we generate a new tumor. This is a new tumor generated after 40 days. Then we track the growth of the new tumor. We show that the new tumor also being suppressed. So that means the T cells there, once activated, there's a memory effect. So to study safety, we actually did the biodistribution studies of the nanoparticles. After injection, we waited for 16 days. We want to see where the nanoparticles are in different organs and tissues. So to our surprise, typically there's a pretty high accumulation in the liver, but not in this case. You can see the blue bars are the controls. So actually a lot of nanoparticles for some reason went to stomach. We are still trying to understand what's the reason. So we also did toxicity studies. We show that with the nanoparticles with APH in different tissues like heart, lung, liver, spleen, and kidney, we did not really see any indication of toxicity. So that means this treatment should be safe. So one question is clinical use, right? Clinical use. Because we use magnetic nanoparticle heating, it requires a machine to apply the alternating magnetic field. Fortunately, there are companies that have been working on those. In particular, there's a company called MagForce. They have produced machines like this. Actually, it's a large scale. You could have patients slide into the machine just like MR scanner. And they have done clinical trials in Europe, and now they are doing clinical trial in the US. So from device point of view, this is entirely viable. So now let me switch gear to genome editing. As you may know, genome editing is becoming a very big field. So the idea is to precisely modify a genome. The question is how, right? So we generate engineered nuclease. In the older days, it's a zinc finger nuclease. Now we use CRISPR-Cas9. So the engineered nuclease generate a DNA double-strand break at a defined location. That's very key. It's not random. You design where the cutting occurs. So once that happens, cells have two repair pathways. The first one is called non-homologous end joining. That actually can rejoin the broken end very quickly, but in doing so, the cells make a lot of mistakes. So that's why it's called error-prone. But that could be useful because you can use this to disrupt a gene, or you can generate large gene deletions. The second pathway is called homology direct repair, or HDR, that requires a template. If you supply a template without mutation, you can actually fix the mutation. That's called gene correction. Or you can do large gene insertion. So for therapeutic purposes, there are two formats. One format is take cells out from patients, deliver the gene editing machinery into those cells, then fill the cells back. Okay, so that's one format that actually most of the clinical trials now using gene editing follow this format. So in vivo gene editing actually is very important because a lot of cells you cannot really take out. For example, motor neurons, right? So you have to deliver gene editing machinery directly into patients. This, I believe, is the future. So for sickle cell disease, as you may know, is due to a single mutation, 80 mutation in beta globin gene. In the world, there are more than 20 million people having sickle cell disease. In the US, it's about 100,000. In the world, actually, every year, more than 120,000 people die because of sickle cell disease. In fact, a lot of patients are in Africa and in India. So treating this disease actually could save a lot of lives, especially a lot of infants are born with sickle cell disease. In fact, in Africa, 90% of infants with sickle cell disease die before age of 5, right? So it's a very big disease. There's no cure except bone marrow transplantation. In fact, that requires a matching of genotype. So in theory, only 15% patients could have that. In practice, actually, only around 3% of patients can be treated by bone marrow transplantation. That means there's no cure for the majority of patients. So over the years, there are quite a few approaches developed using gene editing for treating this disease. For example, the idea of disrupting a particular gene segment called BCL11A enhancer in patient cells. That one has been advanced through phase 1, phase 2, phase 3 clinical trials. In fact, I will show one slide. Tuesday last week, just a few days ago, FDA discussed the application by Vertex for clinical use of this method. So the second approach is to direct correct the sickle mutation. There are two approaches. The one is to use the long donor packaged in AAV6. The other is using a short donor. The third approach is to do base editing. That is, you only modify one base instead of generating DNA double-strand breaks. Okay, so our approach is to do gene editing with a very short donor. So again, the idea is to use CRISPR-Cas9 to isolate cells from patients, deliver CRISPR-Cas9 with the donor template for gene editing, then deliver the cells back. So a lot of steps in the clinical practice. One key aspect is the following. For ex vivo gene editing, after you take out a certain amount of patient stem cells, the doctors will need to damage all the remaining HSC stem cells by using either chemo or radiation therapy. That actually increases not only the cost, but also risk to patients. Okay, so ideally, actually, we would not need to do that. I will touch upon this point later. So we have done a lot of studies over the years. We systematically optimize the CRISPR system. We optimize the donor template. We deliver the CRISPR-Cas9 system using electroporation. That's ex vivo. Then we quantify the editing rate. We quantify the off-target cutting. We also performed engraftment. So just show you some data. This is the early data we have. This shows the gene correction with 5 patient cells. So each dot shows the result from a particular patient. You can see we can achieve between 25 to 40% of gene correction. And we can also have cutting, but without gene correction. Here shows there's no cutting. So if we perform gene editing, then differentiate the stem cells all the way to red blood cells, you can see without gene editing, roughly 20% of cells are sickle cells. With gene editing, only 1.3%. Okay, so this shows the promise. At the molecular level, we've shown that the red is actually the sickling hemoglobin. Without gene editing, this is actually a patient under treatment. You can see dominantly it's a sickling hemoglobin, right? So with gene correction, the red part shrinks dramatically. The green part, which is normal hemoglobin, increases dramatically. Also, with just cutting, the blue part is the induction of fetal hemoglobin, which is actually beneficial. So we have done a lot of off-target analysis. I don't have time to go into the details. We developed a software to analyze, to identify potential off-target sites. We did a lot of experimental validation to show that if you have off-target, in this case, actually on-target cutting, off-target cutting are in the same chromosome. That actually induces a large chromosomal inversion and a large chromosomal deletion. So we performed engraftment, initially 2 engraftment studies. The first one is to use patient cells from peripheral blood. The second engraftment, we use cells from bone marrow from patients, both successful. Then a few years ago, we worked with a team at Stanford to apply for clinical trial, and we generate additional data to show the method works. In fact, there are 2 clinical trials using the CRISPR system we designed. And the first one, again, led by my collaborator, Matt Porteous. the clinical trial has been conducted by a company called Graphite Bio. The second clinical trial using the same CRISPR system, but with a short donor led by Mark Waters at UCSF. Okay, so the clinical trials are ongoing. So there's a recent issue. So in the whole gene editing field, typically people quantify the outcome gene editing by detecting the small deletions, small insertions, so-called small indels. Those are usually 20, 30 base pairs. Now, recently we found there are large deletions larger than 200 bases, the large insertions larger than 50 bases. So if you do the typical quantification, you would miss all of those, okay, because those are large. So we did a different type of sequencing called SMART, single molecule real time, which also together with a so-called UMI, that is unique molecular identifier, because in this case we have to use long-range PCR. We found that in this particular case, more than 35% of large deletions, and we also had intermediate deletions. We had close to 2% of large insertions. So those actually are at the on-target side. It's a new issue for the whole genome editing field. So large insertions up to 1,000 bases actually came from different chromosomes, right? So we are still trying to understand why we have these large deletion, the insertions. So not only the particular guide, a lot of different guides shown the same. In particular, the RO2, labeled RO2, that's the guide I just shown in 2 clinical trials. And the BCL11A guide actually was the one used phase 1, phase 2, phase 3 clinical trials. And all of those show large deletions and large insertions, okay? So if we do animal study, we show that after 16 weeks, those large deletions are still there, okay? So they persist. So we have a lot of unknowns. What's the mechanism? What are biological consequences, and how to address this issue? As I mentioned, just last Tuesday, there was a whole day meeting at FDA to discuss the application by Vertex for using gene editing to treat sickle cell disease. The good news is that they tested cells from 30 different patients. All of those studies show there's a sustained induction of fetal hemoglobin, about 20% for at least a year, right? So that means the efficacy is pretty good. Now, there are concerns. One concern is that the company only did very minimal off-target analysis, only analyzed cells from 3 patients. In fact, in our paper published a few years ago, we analyzed cells from 7 patients. Why in phase 1, phase 2, phase 3 clinical trials, they only analyzed 3 patient cells, right? So that's an issue. The second issue is what I just mentioned. There are unintended large gene modifications at the on-target side. In fact, at this meeting, Dan gave a very nice talk, overview of the work on the off-target and on-target analysis. He's shown in his slide 19 our results, right, to show actually this is a new issue that has to be addressed. So another very important issue is cost, right? So how much this will cost? The estimate is that is at least $2 million per patient. In fact, some people even predicted will be like $3 million per patient, right? So with this high cost, there's no hope to treat a lot of patients in Africa, also in India, in many places in the world, right? This is really a big issue. So to reduce the cost, the only way, in my mind, is to do in vivo gene editing. That is, you directly inject the CRISPR-Cas9 into patients. So the hope is that each injection will be just a few $1,000s, right? You can do multiple injections, but the big challenge is efficacy, because there you need to show there's a high enough efficacy because you do not really touch bone marrow. You still have a lot of stem cells producing sickle cells, right? Unless you can achieve a high level of efficacy, otherwise this would not work, okay? So then delivery is a major engineering issue. How would you deliver CRISPR-Cas9 into stem cells? Using viral system like AEV, you may have actually immune response, okay? Also there are genotoxicity. You use nanoparticles efficiency is low, right? So there are a lot of studies going on in this area. So in summary, we analyzed the site dependence of magnetic nanoparticles to demonstrate the classic theory actually is not correct for large nanoparticles. We use nanoclusters together with AEPH to show that we can treat cancer metastasis. That's the potential. We also developed gene editing-based approaches for curing sickle cell disease. The preclinical results are very promising. The clinical trials, the 2 clinical trials are underway. But a new issue is unintended large gene modifications, as I just shown you. We actually need to address this issue in order to make sure it's safe to do gene editing for treating sickle cell disease. I would like to acknowledge my lab members. So the first part was due to a few postdocs together with a graduate student. The gene editing work also was due to multiple postdocs with my graduate students. I have 2 collaborators on gene editing, Matt Porteous at Stanford and Vivian Xian at Emory. With this, thank you so much.
So thank you very much, Dr. Bao, for such a wonderful talk. And we have time for 10 minutes now to ask questions. So the floor is open for the question.
Gang, very nice presentation. Thank you. So I have, in your AAPH treatment and then using alternate magnetic fields, is it not harmful to your tissue microenvironment, which includes infiltrating T cells and other cells? Don't your free radicals kill those as well?
Yes.
And so how would you ameliorate that in your treatment? You would not want to kill normal cells, for example, around that region.
So that's a great question. In this case, actually, we inject nanoparticles directly into tumor.
Right, but they're infiltrating immune cells in your solid tumor. And so these infiltrating immune cells like T cells, CD8 T cells, which you showed, are actually beneficial in various ways.
Right. So the truth is, if you have nanoclusters with AAPH, once you apply magnetic field, you can actually kill all kinds of cells, right? But we believe predominantly, we actually did some analysis to show predominantly those cancer cells. Why? Because cancer cells are more sensitive to high temperature. That's actually the basis of using magnetic nanoparticles to do hypothermia therapy, right? So that's why actually we mostly kill tumor cells. But that's a great question.
I am very intrigued by this large piece identification in the genome editing. So, but then along the end, you mentioned that the only way to bring down the cost is to do in vivo genome editing.
Yes.
And if that were the case, has there been any thought on how to take care of these small and large pieces if we're going to do in vivo genome editing?
That's a great question. Let me just go back a few slides. So here we show, in addition to delivering CRISPR-Cas9, you deliver a short donor template. In this case, actually, the 121 bases. if you deliver that, actually you shift the repair pathway from niche jet to HDR. So the instead of having 35% of large deletion, it became less than 9%. Okay. So in my mind, this is a way to dramatically reduce large deletions. It will not eliminate entirely, but you reduce that, right? So we are still doing a lot of study to say, if you have large deletions, what are the biological consequences, right? So one immediate consequence is that due to large deletions, the beta globin gene will not be expressed, right? So that is one risk. The other risk is you have truncated proteins made due to large deletions, that those could be toxic, right? So the biological consequences are still unknown, but we hope that by reducing a lot of deletions, you dramatically reduce the risk of those. That's a great question.
Thank you.
Thank you very much. Very interesting, actually, both the sections of your talk. So majorly my question or maybe a curiosity is from the point of view actually which you mentioned about the tumor metastasis. So the model which you were describing here actually, and you see actually the effect on the secondary tumor. So is it in the real situation actually that model is something very close?
Great question. Actually, the answer is no. So this is really the first model. We use a colon cancer model number one. Also, this is the tumor generation is using sub-Q, right? Both actually having issues. Why? Because the colon tumor, in this case, is known to be immunohot, meaning if you use nanoparticles, you can effectively generate T-cell response. So the question, what if the tumor is immunocold? For example, it's well known that the tumor of pancreatic cancer is immunocold, right? So also the second part is the sub-Q model is not the best model. And now we are using orthotopic model. So after we published this paper a year ago, we started working with my colleagues at MD Anderson to generate, number one, pancreatic cancer model, which is again immunocold. The second is to generate orthotopic tumor in the liver, because this is pancreatic cancer, the metastasis usually is in the liver. So we are doing both now, but it's definitely a great question. So with that, I hope we have the models closer to clinical situation.
Thank you so much. Thank you.
So I have one question. So it's on magnetic nanoparticles, which you said that each cell will have around 50,000 nanoparticles accumulated. So while you have shown the efficacy and also that it is not toxic, but what is the fate of those nanoparticles eventually? Are they staying there, or they are dissociated or metabolized? Did you get a chance to look at that?
So great question. So we have done some studies. Other groups also have done studies. So the reason we chose iron oxide nanoparticles is because in the end, when cells die, the nanoparticles are being released. So nanoparticles actually either being removed from the body or they accumulate in the liver. In the liver, they are being decomposed into iron and water over time. So therefore, the magnetic nanoparticles are non-toxic, also already received FDA approval for human use. And
another question is on regulatory bodies, recognition that CRISPR-Cas will be now a future technology. So where FDA is right now in terms of developing regulatory guidelines for such a technology? Yeah, great question. In fact, I was involved in some of the FDA discussions. So the efficacy actually is clear, it's there. So the most of the discussions
were about safety, right? So especially off-target cutting can generate tumors or tumors or other diseases. But to be able to tell, you need to follow the patient for many years, right? So those clinical trials are just over the last 3 years, right? So that was a debate actually even last Tuesday. I actually watched part of the meeting in real time. So some people said you need to follow the patient for 15 years. But the debate is that, do we want to wait for 15 years to approve the clinical trial, or we give approval, right? So there's a risk and a benefit consideration, right? You need to consider both, right?
Great. So, yeah.
Thank you, Dr. Bao, for the great talk. So actually, I was interested in the approach of using long read sequencing techniques for the large insertion, especially.
Yeah.
So in the slide following this particular slide, you showed that it follows from particular chromosomes. You see there is a pattern.
Yeah. Let me go to that. Yeah. Here. Right.
Yeah. So do you see any the sequences which are getting inserted? Are those stochastic, or do you see any pattern in those? And also, do you speculate that those large insertions that are being incorporated, they may have some particular features, for example, activation of some retrotransposable elements in this particular area that it is being inserted?
Yeah, those are great questions. In fact, we analyzed the inserted sequences, right? So actually, first we saw those sequences may have similarities with the Kasai sequence, right? Actually, that's not the case. We saw all kind of sequences being inserted, right? So the question, why? Why those sequences are from different chromosomes, right? So one possibility we have been looking into is that because there's a 3D structure, right? Those could be the locations in 3D space. Those particular locations are closer to the Kasai. That's why we have pieces of DNA being inserted there. For now, really just a hypothesis. We are still doing some analysis to show if this is true or not. But that's a wonderful question.
Okay, so I think we are done with the questions. I would like to thank you, Dr. Bao, for such a wonderful presentation. So our next lecture will be delivered by Dr. Venkataramanan from Karkinos Healthcare. He will deliver the Aurobindo Nath Bose Entrepreneurship lecture, and the title is Molecular Medicine in Oncology Practice. Dr. Venkata is founder and CEO of Karkinos Healthcare, and he's an advisor to the chairman Reliance Industries. Prior to founding Karkinos, Venkat spent more than 15 years with the Tata Group in various capacities. He was a member of the governing board of multiple institutions such as Tata Medical Centre, ISC and TIFR. He is the executive director and CIO of R&T Capital Advisors. Venkat is a science and law graduate and has done his master's in business administration. Dr. Venkat he's completed his doctorate in engineering from University of Warwick. Venkat is also a member of Governing Council of Geo Institute, a multidisciplinary higher education institute of eminence. So with this, I invite Dr. Venkataramanan for delivering his lecture.
Thank you. Good afternoon, everyone. Coming to an IIT is often quite intimidating. That too when you flunk JEE a couple of times and much later in your life and career, not only you're asked to come here and be a faculty and not also sit in front of incredibly smart minds and try to convey what you're attempting to convey. Thank you for your being here, and afternoon it's quite tough to be awake. And for me as well. So in that sense, we'll try to lighten it up and come up with a few anecdotes. Much of what the earlier speaker said was way above my pay grade. So much of it is Greek and Latin. I'll start with Greek. Karkinos is a Greek acronym for a giant crab, which the legendary health tech entrepreneur in India, Dr. Pratap Reddy of Apollo, said, unless you talk Greek and Latin healthcare, nobody takes you seriously. So for us, coining this name was a double whammy. On one hand, being a private enterprise, you're always running after cash from potential investors with a large begging bowl or chasing revenues. So when you have such a name, people will say, "Arey, iske andar kya hai?" When you not open the slide and see, otherwise it just gets passed on to anything else. On a more serious note, this is also the word which Hippocrates in medicine coined this for cancer in India as well. The symbol for giant crab is a Karkata, the Rashi for cancer. I was fortunate and privileged to spend almost 20 years in the Tata Group, 15 of which I was closely associated with our former chairman, earlier as his chief of staff, and or subsequently when he claimed that he retired. People like him don't retire. So I ended up in his office as initially as a trustee, then had a chance to run the trust as managing trustee for about 7 years. Again, thanks to that, for many of the iconic institutions which I could never get admission, like a TIFR or ISC or TISS, ended up as a trust nominee on those institutions, signing what you call certificates for potential students passing out. So luck can come back to many of you all in some other form or the other, or institution that you couldn't get to. One great inspiration from him has been, thanks to him, one has had the privilege of meeting several iconic people in India or abroad, ranging from President of United States, not the Trump, but many others before him, George Bush and Joe Biden earlier, as well as the chief of Mossad. So the diversity of experience is quite varied, and in many ways, again, thanks to him, one had seen many iconic or currently iconic entrepreneurs or industrialists who were in their earlier days had a few traits of passion, curiosity and conviction to do something which, when people say seemingly impossible, you attempt at least 80% of it, at least you can get there. So some of those design principles one has attempted to apply in our current journey of what we're attempting to. And whatever I say or speak is all the good things are mine, all the bad things are my colleagues. But on a more serious note, this is a journey and a manifestation of several years or decades of experience of multitude of people who have been struggling to find a potential solution to a seemingly intractable goal. And this is a work in progress, by no way perfect, but we do believe we have the building blocks of a journey which potentially can unleash many more innovations going forward. The challenge in India, unlike many other countries in the West, is we detect cases quite late. 70% of cases in cancer are what we call stage 3 or stage 4, and 90% of that 70 end up effectively like a palliative or an end of life case. While we have 200 odd cancer centers, the top 60% are in the West and South. There is a significant asymmetry in geographic distribution as well. And the reason of picking this up was Tata's as an institution had been pioneers in oncology, having set up Asia's first cancer center in 1930s, which is now the Tata Memorial Hospital, taken over by the Atomic Energy Commission. And over the years had helped number of non-profits to come up with potential solutions on either donating equipment, funding cancer research, or establishing other organizations as well. So while it's very clear that there were other two sub-challenges as well, while we all say early detection saves lives, but even within a comprehensive cancer center, there are multitude of practices which people often struggle to come to grips with. In a sense, many a times when in India, almost 80% of the procedures is due to a surgical protocol. If you go and see a surgeon, he says, okay, I've seen it, chopped it off, wait for 6 months, take a PET CT, then figure out what needs to be done. If you go and see a med-onc, depending upon the type and kind of work that he or she is familiar with, gets onto either a immunohormone therapy, XYZ. Same is true with the radiation oncologist who will say the treatment in the machine I was trained or what vendor familiarity I have, that's the best thing, so to speak. Decisions are often not taken on the back of a tumor board based on an information sharing hierarchy. So there were a lot of systemic inefficiencies we noticed, which we couldn't quite address in a conventional brick and mortar edifice as a standalone single institution entity. Therefore, by design, we said, can we conceive of something which is participatory, not proprietary, shareable, not ownable, and with a view that will not be perfect, try to look at what you call a least common multiple of an association rather than a highest common factor of an engagement in terms of a contract and or an agreement. That was the genesis, so to speak, of this journey. Again, we are woefully short of infrastructure. If we take radiotherapy as a corollary to access to cancer treatment, almost 50% of cancer patients undergo radiotherapy in some form or the other during the journey. There's a recent report of Ernst & Young. US with about 1/3 of the population of India has close to 9,000 linear accelerators. China has close to probably 7,500. India, the last count was 714. 2 years ago, it was 684. China is adding about 350 a year. India is adding 50 a year. But at the same time, we don't need to be quite intimidated by this, because we have seen those trends play out in banking, fintech, steel, retail, etc., where such exponential growth and transitions have indeed happened, provided someone took that leap of faith to challenge many of the notions and conventions. Again, cancer also is like, as earlier speakers and others talked of, a much more scientific protocol. We still don't know a lot of it, and despite the best of advances in medicine, many of the insights which people have historically generated have all been based on American and Caucasian population, not necessarily or Asian or Indian. And where in countries like us, where affordability and accessibility within a same geography is quite startling. There's a city of Mumbai where on one hand you find skyscrapers, just next to it you find slums, and this is true across most modern metropolises across the globe. Paris has the same issue, Sao Paulo has the same issue, and even some of the states in the United States also have a same issue. So our notion was, instead of looking at a cancer center as a standalone brick and mortar edifice, can we conceive of a establishment which effectively becomes, like my co-founder calls it, as a community as a cancer center, where there is a shared responsibility of everyone in the ecosystem and not just one individual or entity. Even recently, Mayo Clinic is trying a similar enterprise which they call as Cancer Care Beyond Walls. And the reason of attempting this is, like say, how can we integrate an otherwise distributed care ecosystem riding on existing distribution networks, like say when we take to early detection using even conventional tools. I'm not getting into liquid biopsies or Grail type of tests yet, which we can do probably at a much fraction of the time and cost, which we'll talk about it in some time. But at the same time, even using conventional healthcare networks, because of the significant information asymmetries that exist, we often miss it out. I'm sure it happens even within departments or faculties or XYZ. Many dentists can pick up precancerous oral lesions. Many gynecologists can screen and treat early stage cervical and or breast cancer. Many pulmonologists can pick up potential signs of a lung cancer, except they misdiagnosed as a tuberculosis or a chest infection, so to speak. So in that sense, if we are able to detect early, get the diagnostics right, that too instead of a traditional morphology-based characterization of diseases, to molecular / next generation sequencing and hopefully whole genome and whole transcriptome sequencing as well, where delivery happens, because you're unbundling the knowledge and delivery can be taken at least for common cancers. One is not talking about the complex gallbladder, soft tissue, or many more advanced cases, but breast, cervical, head and neck, oral, lung, colorectal, which are in any event 65 to 70% of any low and middle income countries' cancer burden, can easily be taken close to a community. The discovery part of it as well, if we are able to integrate that piece on the back of a technical input by virtue of doing the clinical practice with the degree of an academic rigor, at the same time have the mindset of a researcher in perpetuity and a seeker of a knowledge in perpetuity, perhaps we might come out with something which might potentially be spectacular. And the origin and premise, as I mentioned, was one of the key learnings we had was in Varanasi. We were asked to build a cancer center in Varanasi. Don't ask me why Varanasi. That's a story and a debate for another day, because two cities which cannot be named, Varanasi and Ahmedabad, nobody can say anything or comment anything. But we were humbled to be part of that journey of being allowed to explore the opportunity of commissioning 1 million square feet of cancer center in less than a year. The normal timelines were between 5 and 7, because it so happened in events and circumstances, a foundation stone was laid and 2.5 years nothing happened. Suddenly somebody woke up and said 2019 is coming, what do we do? So me and a few colleagues of mine, Lakshman was an IIT IIM grad, we're sitting in a trustees meeting. They said, if you give us an opportunity, we'll try. We went ahead and did, not knowing what it takes or no background or knowledge on what can be done, but we completed that in 11 months flat, which got the record for the fastest ever completion of a tertiary infrastructure by a charitable institution. And in the very first year of operations, a facility which has 12 operating theaters, 7 linear accelerators, a cyclotron, saw new registrations of 20,000 people. There's a waiting list of 8 weeks for radiotherapy, and the load in Bombay hasn't come down a bit. So we had done a similar initiative in Assam as well, where in partnership with Government of Assam, my previous organization conceived a distributed model of care adjoining a medical college rather than a standalone center. So we see that India is a significantly supply constrained environment, provided the supply is accessible, credible, on the back of a trustworthy knowledge protocol. There is there and or the market for the taking. And as mentioned earlier, this is still vastly unknown. This is a paper by Vivek Subaya earlier this year. Despite all the advances in medicine, especially in oncology, almost 80% of it is still unknown. And much of it is, again, a couple of years paper. It's like whatever you do, it comes back like a monster, as they call it, as a 9-headed hunter. I don't want to get into the technicals of it, but the important factor of it is we missed this trend in trust when we were looking at a traditional modality of engagement, and why the reason why one is too much emphasizing on molecular. I'm more mentioning from a pragmatic example. Don't hold me on to the science part of it. I don't know the C or S of it, as the case might be. Only tell as an illustration, when we were in San Diego about 12 years ago, Mr. Tata had the benefit of taking his whole genome and whole transcriptome sequencing from Craig Venter's lab. It cost him 30,000 US, and the reports came in about 10 months, right? Today you can do that at probably $1,000 or less, and you can get a report in a clinical setting, as Memorial Sloan Kettering and others have demonstrated. There's a paper by Neil Shukla last year. In 9 days flat you can do at a price point of $1,000. It literally in less than 10 years time. What it also demonstrates is, when many of the targeted therapies which have come in, especially in the last 3 years, none of this have been based on any Asian or Indian population. Whereas when it comes to market authorizations, we are also not part of the global phase 1 or early phase trials whatsoever. So for us, therefore in India, where we do believe is a tremendous opportunity, especially with institutions like the Mehta Center for Engineering and Medicine coming up. We can, in Telugu, say, late go chna latest go quarterly. I'm talking a local language. And just because you're late, it doesn't preclude you from being the latest. So we can literally leapfrog a generation in terms of infrastructure, facilities, and capabilities, as we have demonstrated in telecom. Just to give a example, in Tata's, we had a company called Tata Docomo. Where we used to celebrate when we used to get 200,000 subscribers a month. There used to be parties in Taj, 2 lakh people, great, etc., etc. And 70% of the revenues that we used to generate were on voice telephony. They came an operator in 2016, completely redefined from a conventional 2G to an all IP network of 4G, and said voice is free. And the first 100 days, they gathered 1 million subscribers a day. So just changing or framing the zone or frame of reference in terms of even when we were conceiving, Reliance also is an investor. Shambhani said, solve a problem at scale and money will follow. If you chase money, neither you'll solve a problem nor make money. I've done this for 40 years and I've never failed. Second is that as a budding entrepreneur, you guys chase what is my valuation in series A, B, C, D, X, Y, Z. Try to drive value for people. Valuation is a substitute and a corollary for valuation. Try to do anything in between, you'll succeed, no problem at all. So when we apply some of these design principles to health tech or biotech, for us what currently is a crisis by virtue of some of these things not being here, if we are able to create a mind space of a generational shift in capacity, generational shift in the need and rationale for molecular diagnostics upfront, before beginning first line, already it's happening in several forms of cancer. Like say Australia does whole genome and whole transcriptome sequencing for all pediatric patients before first line as a national case. Whereas we are still struggling by 5 gene, 10 gene, it's kind of completely irrelevant. Today, in this day and age, that too when many of these have not been relevant and or applicable for Indian cohorts in any case, why the heck we are wanting to look at previous generational constructs. And as the earlier speaker also has mentioned, this is not necessarily a one-size-fits-all approach. So it therefore needs constant integration, assimilation, and today we have a number of drugs which are kind of pan-cancer type of markers as well, not necessarily a unique original site of origin as they came or claim to be. And instead of therefore a one-size-fits-all approach, can we look at the opportunity of not just precision medicine but also personalized medicine? Because any type of tumor that you take, we are globally number 1 or number 2. We have the dubious distinction of it, and things like head and neck. I guess Kanpur has a phenomenal role to play in the propagation of head and neck, thanks to many of the factories, tobacco, pan masala, and all sorts of things that exist out here. I think for us, we also would need to take a public health approach to some of these chronic diseases rather than assuming that it's a government responsibility alone. And we have also seen from a patient outcomes, many of the most advanced immunotherapy protocols for an Indian cohort, the applicability is less than 14%, whereas normally left, right and center is prescribed, take Pembro, and we also have some studies to demonstrate low-dose immunotherapy trials work equally well or better as the case might be elsewhere, but because of some of the interests or vested interests as the case might be, we often are not able to get access to it at that point of time. And this is also not currently translated into a regular clinical practice because all of this since historically have been in silos. As mentioned, 90% of first-line treatment happens based on histology alone. Less than 15% do IHC. And IHC also only a few labs do IHCs much better than most because it's a highly skill-based assessment. Molecular is less than 5%. So instead of focusing on conventional wisdom and or ecosystem, can we work with leading academic institutions to integrate these workflows in a way that on one hand solves a diagnostic issue, on the other hand also helps in ensuring a patient navigation. In fact, my first recollection of someone like the Mehtas existing was when we went to IIT and they said they have a lab and it's in the Mehta School of Biomedical Sciences or Engineering, and said they had all the best of equipments but very limited clinical input of what needs to be done. They were chasing clinicians, whereas we were chasing for good scientists and researchers, and they were sitting on at the National Cancer Tissue Biobank for almost 10 years. I wouldn't comment on what they did or not did, but we said, look, let us write up a detailed protocol where it benefits on one hand and solves a clinical problem, addresses your research questionnaire. You only be the principal author or the prime author and get whatever your impact factor or H index up or down as the case might be. But ensure it is applicable from a relevance to a patient perspective rather than being too puritanical as often we tend to get into in such leading academic institutions, right? And to their credit, they were quite supportive in such response. We then added a few equipment to the lab to ensure the diagnostic workflow and the clinical and the research workflow is all combined, put in our own people as technicians for pathology, and since IIT is a research institution, negotiated with the dean, school and faculty to say that, look, in the case of there is a money exchange or revenue, what is the share of XYZ can happen. So it took us almost 9 months to crack the first contract, but once we did, we managed to more or less replicate it across most technology institutions as to how this goes along. But those are first experiments within an IIT, within a department. We realized that, I'll come to that in a moment, but there are still several pain points in the mix. In small towns and villages, the reach is quite limited. Several of the low-income patients have a phenomenal barrier to entry, so to speak, and the awareness itself is restricted to top 10 or 15 cities, as the case might be. What we are therefore attempting to is taking a leaf out of the telecom story or the whole genome story. Can we literally democratize the entire approach to get to where we have to at the fastest level of time, as the case might be? And this is indeed possible. It's like, say, we can, 15,000 can, if luck goes, we can probably get to 5,000 or less. Probably the initial adoption might be quite tough. That's where with partners like yourselves, can we, because researchers are always chasing for samples, clinicians are always looking for insights which a researcher can potentially give, not necessarily with a hypothesis, but with a degree of certainty. Because end of the day, a patient comes to a clinician. This is what Mayo has taught us brutally as well. You might be how great a researcher or how great an administrator or a financer. It's a dyad, doctor, administrator. So the trust of it you can't take away. Though we might claim that doctor doesn't know anything, I've done 20 years of research, you should try it out. In clinic, on a day-to-day basis, it just doesn't work. So if we have the humility to engage and solve his or her problems, I'll come to on how they've helped us to develop the technology in a way, then they'll be your best friends in terms of either giving access to a tissue, reporting patient outcomes, because whether we like it or not, they control the front end of the patient, period. And taking a leaf out of this, we created a network of advanced molecular diagnostic labs. The one in Lucknow also is in partnership with IIT Kanpur and the Kalyan Singh Medical Center. In this also, for us, it was a learning. In our first lab, we thought by creating a lab, samples will come. Second lab, we thought by being in the biotech department, samples will come. Third lab, we said, if in a research institution in a state which has a cancer centers connected in Assam, we thought it will come. Fourth one, we realized unless you are next to a clinic and within a clinic, getting fastest access from the operating theater is the best way to get, rather than trying to hold on to what I do or don't do. So for us in the last 18 to 24 months, it's been a learning and iterative process, even in terms of execution, as to what is the best and the right thing to get access to the frozen section upfront, so that the research rigor that we can maintain at the time of solving a diagnostic output, because cutting the tissue and getting an FFP block is a 100-year-old technology. If we get a frozen section and put straight into molecular characterization on a WGS or a NGS, the outcomes obviously will be much better, provided a clinician there also exists to tell and certify the quality of tumor content upfront. So these have been some of the learnings in terms of our execution on the ground. And the things, it's the Lucknow lab should be ready probably by early January, or definitely by early February. Ourselves curated a range of diagnostic techs. Some of them are in work in progress. We have all the national level approvals for it. We'll also have the CAP clear approvals as well by the next quarter. So we can hold ourselves to any large US academic center lab. These are some of our panels that we have. I won't get more details into it. But at the end of the day, we are an entrepreneurial commercial organization. We don't have large donors, or our mandate, while we are a for-profit organization, the mandate given is be sustainable. Then only your rate of return can get your weighted average cost of capital. And for many academic institutions, that's a very steep climb, because you're tenured, you have a lot of people from government grant giving departments. Sir, grant lik dijiye sir, mera 31st expire ho raha hai, please. You all, once we reach a particular stage or stature, we all know as to how the grant system and the different institutions literally love to give to institutions like IIT Kanpur, because they are de-risked. If they would IIT Kanpur, nobody will ask them any question. But when we really look at the commercial and or the translational rigor of some things, like say how many researchers and or clinicians really know how to write a R01 or a U1 grant, when we benchmark ourselves to the best of the NCI related process of holding ourselves accountable to the highest standards of rigor. Maybe in places like IIT, many might have papers in a New England or a Nature, but for many, one paper in their entire lifetime, they feel they've arrived in life. But whereas when you go to Mass General or places like this, people have every 3 days or 4 days, something or other coming up from their PhD or a postdoc, as the case might be. So in that sense, this partnership with institutions also is increasing. And since our notion of community as a cancer, we have done close to 22,000 tests. In a state like UP, we can do 22,000 tests in probably less than 6 months instead of 2 years or 3 years, as the case might be, because entire UP itself is more than all of Europe combined, just from a scale perspective. And rather than trying to restrict ourselves to the top medical oncologists who at times claim they know it all and have found a way to solve it all, try to work with the long tail of people who are hungry for new generation knowledge and who are really at the cutting edge of the initial level of a patient engagement to pick up those signs and symptoms. And this entire unbundling process can play a key role, at least in addressing some of the common issues upfront. And we ourselves during this process discovered a number of things in this. Say one example, my chief scientist can explain it more clearly. We found HPV testing was regularly done. Normal pick up in a community screening, pick it up, do what you want. But since during this process, we embedded the rigor of academic vis-a-vis research combined. So we had a bunch of what you call biologists looking at what in those 250 or 300 odd cohort in partnership with some of the cancer institutions saying, what is it? So they noticed a particular protein expression, STX3. And again, there was a chemistry professor in IIT Gandhinagar who said, look, I have 17 compounds. Since we had banked the cell lines and had it in our lab, test it out. And 2 of the 17 work. Since it was coming from a clinical input first, the ability to take it to pharma and or diagnostic agencies for building specific diagnostic assays becomes much faster as compared to discovering something and then going to a clinician to say, look, I've discovered this, why don't you try it out? We have worked enough with clinicians that doesn't work that easily. So flip that pyramid inverted and try to say, like say when the kings don't give their hand, say take your hand, probably they'll take the lighter win. So, and we also have seen India itself is in abundance with several of the companion diagnostic tools that might exist, which India itself has multiple pharma entities which are wanting to become life sciences and quote unquote biotech powerhouses, provided researchers like yourselves are. Lawrence Labs is a great illustration of you attempting it, but we're only beginning that journey. The opportunities are quite immense once some of the infrastructure that you see out here coming up pretty soon. And we ourselves are, it's also like say working with public policy school. End of the day, state is a very important player rather than trying to get them at the fag end of it, get them involved right up front. So it's not that as if they will object to it at some form or the other, but having them as a policy, this is an example in Impal, we have done that in UP as well, to ensure adoption of whatever we do, where the reimbursements, the knowledge asymmetry, all of it from a payment perspective also happens quite seamlessly. And this is quite economical as well, in a sense, unlike the US where we can't spend billions or trillions as the case might be in healthcare. The cost of healthcare, if we do molecular characterization upfront and take the delivery through a community center, there are enough and more research articles to indicate that it is indeed possible. And there are already enough economic analysis of it as well on the impact of NGS vis-a-vis single gene. Now, there are a lot of studies which are now attempting whole genome and whole transcriptome and long read sequencing, methylation, whole things are coming in. So in order to connect all of this, what we realized was you need an underpinning of certain technology elements which act as a operating system approach to oncology, akin that of an Android rather than a hardware. And we took a leap out of fintech. Multi-trillion transactions happen overnight with that same degree of security, consistency, practices, traceability, etc. When it comes to healthcare, we are often spooked by patient data, privacy, but these have been solved in other industries, including in aviation, where the notion of a checklist and or the air traffic controller, irrespective of whichever geography you are, it's Alpha, Bravo, Charlie, Delta, Echo, Foxtrot equivalent. You can do many of these quite standardized as compared to what you would have not done elsewhere. So we attempted to take a platform approach to things, ranging from the initial risk assessment to early stage screening to diagnostics to ensuring tumor boards are virtual, where you get access to that expertise, not the physical presence of an expert, and connect all of this through a constant patient continuum, so to speak. And we built multiple applications like any other software or a tech entity would do. A front-end application, a person using an assisted screening application, a diagnostic and treatment application. It took us about 250 odd engineers and computer scientists working with 150 plus clinicians telling us what to do, not an engineer developing it and then giving it to a clinician as an iterative process. Both of them drove each other quite mad in the first several months of journey. But we can say with some humility, the damn thing does indeed work. So it's not a theory anymore. It has been deployed across 1.6 million touch points and has a lot of analysis for standardization of care based on the geography, ethnicity, and care continuum, as one might see. And we have also, since it's a white labeled one, irrespective of whichever partner wants to take, we can change the front end quite easily with APIs and SDKs. And since many of the people obviously want access to the best of information and knowledge that is getting curated constantly, with our partnership with Mayo Clinic is not just a knowledge partner, but also an investor in us. Normally, leading global academic institutions are only known to receive money. They're not known to give money. This is probably the first time in Asia where an academic institution in the US has actually put in dollars in India and been a strategic investor in us since our inception, and given access to all their clinical knowledge, which we can also translate to Indian languages as the case might be. And this is a case of how that end-to-end continuum actually works at a community level. This is a test case in a place called Pallivasal in Kerala, where an asymptomatic patient was diagnosed with HPV positive, taken to screening across multiple touch points, colposcopy was done, and then post-colposcopy, it was determined, the lab determined it was a case of adenocarcinoma. Surgery was done again in a partner hospital, survivorship managed in a community center. Effectively, 5 or 6 different legal entities came together to deliver care which otherwise she would have got only in a comprehensive cancer center. And all of this data end-to-end is available in a knowledge curated digital first approach. And the STX3 example that you saw earlier was one of these cases which had that access to the tissue. Researchers were able to determine it and then find out what the potential for assays will be or could be. So this has been our progress so far. And the opportunity in partnership with you, it's not just 100,000 plus, probably easily 100,000 plus in less than 3 or 4 years time frame, where India can lead and not follow in many of these initiatives. And if you push the boundaries of what can potentially happen, because end of the day, a clinician, as mentioned, is looking for outcomes, not a hypothesis. So in that sense, look at more translational aspects of it rather than pure basic science things. Fine, it's great. You all want to write a paper in Nature working for 16 or 20 odd years, but end of the day, in places like India, people are dying by dime and dozen every single day. What is the difference we can make to him or her in the most accelerated time frame on the back of knowledge that experts like yourselves have at the fastest time frame that we can get by working with multitude of establishments. So the opportunity is there to build a number of products and or services, medtech products and or services, both on the prevention, screening and diagnostic side, as well as on the multi-omic side, because in a state like UP, where data is the currency, you probably have at the highest levels of opportunity than compared to any other state in the country, or probably in most parts of the world as well. So this platform is already available. It's available white label. Your researchers can rip it up, plug it up, take it out and enhance it in whichever form or manner that you want, which gives the clinical information, imaging info, histology info and sequencing info all combined in one single hierarchy built on completely open source tools. And yeah, this is in summary. We are also working with a number of global leaders in oncology, from the Broad Institute to Science of America Foundation, which is the institution of Eric Lander, Mass General Hospital. Yeah, an interesting story on PacBio. And since we talked earlier on long read, one of our approach or lab got noticed by PacBio people and said, look, I don't believe India can do that. So Stephen Turner, who's the CTO and founder of PacBio, said, look, I want to seed myself. So he visited our lab in India, came for a day just for 5 hours and said, we'll give our new generation Revio system. This is the first in Asia is there in our lab in Bombay, and hopefully we'll get that here as well pretty soon. So the mindset of even leading global medtech players to provide us access to their technologies upfront. So in many ways, there's no reason why some of the engineers here can rip it apart and come up with a version of it in probably less than 5 years or 6 years, like what MGI or BGI has done in China, much faster. So the reason of really getting excited about centers like this is you're uniquely placed and blessed with an incredible opportunity, especially many of the youngsters that I see here. And I was telling one of your colleagues that probably take up courses on the business of science as well as compared to engineering and medicine too. So end of the day, it's also an attractive market and commercial opportunity too, in addition to making a difference to significant number of people. You'll also be glad that, look, in addition to network, your net worth also hopefully will increase pretty soon. Thank you.
Thank you, Dr. Venkataramanan, for such a wonderful insight and showing us a new model for diagnosing early cancer. So the house is open for questions now. Yeah.
So probably in the last but few slides, there was this slides of drug discovery. You can elaborate on that.
See, in this, like say, our entire tagline has been on discovery through delivery. In a sense, let's take a specific example. Like say, if you take PD-1 or PD-L1, when the clinical trials were done in 14-15, out of the total trial of MSD was 248. None of them were from Asia or India. And even if you take the revenues from that particular PD-1, which later translated into Pembro, in 14-15, the revenues from that particular drug were 55 million. Can anyone take a guess as to what the current revenues from that particular drug are for Merck today? Wild guess. As wild as you can be. 29 billion. 29. One single drug, right? And Roche has similar one for PDL1 called Tecentriq, etc. Whereas in India, because of lack of molecular characterization upfront and the ability and 249 people in India, we were doing a similar trial for Rakuten for head and neck. When we had Miki Mikitani, the chairman of Rakuten, did not believe that in their entire 3-year journey, they could get about 80 samples. So when we were talking to clinicians, they said, yeah, 80, 3 months. So he didn't believe. So we got him to the operating theatre at Tata Memorial. They see 49, 50 cases a day. So getting 80 in 3 months was nothing with the same degree of inclusion and exclusion criteria. So he thought we won't have advanced MRI machines. He thought people would not know what an NCCN guideline is. He thought people do not know what a US researcher or an academician will be. Today, our people, many of them have studied there, come back, are equally bright, if not more competent than anyone else. All they need is an opportunity to make a difference on the back of an infrastructure that is created, and more the academic freedom which allows them to pursue an otherwise intractable solution. So I think in many ways, the tagline of our Honorable Prime Minister, this is a very different country. It's an aspirational nation with a inspirational leadership, and in the quest for next 20 or 25 years of a journey, this is attempting several things which are impossible. He himself says, "Namumkin bhi mumkin hai." In a sense, the tagline of many things that he mentions, "Sabka Saath," work with everyone. Not in silos. "Sabka Vikas," work for the benefit of everyone. "Sabka Vishwas," take the faith of everyone. And therefore, you tend to do many things which otherwise you think are just not possible. So even we had a chance to meet him some time ago. I'll just take this, put one slide over there. We had it was supposed to be a 15-minute meeting with him. We ended up spending almost an hour. And his parting comment was, work on things which make a difference to large number of people. So he's not interested in incremental changes whatsoever. So India in that sense gives the land and the potential to explore exponentials. It's not incremental at all.
In your plan for this is kind of like a decentralized healthcare system. How do you ensure ownership of diagnosis as well as trust in decision makers who are not part of your institution?
See, in medicine, only if the Pope says it is sermon. Across every segment of a diagnostic process, there are giants who are very good in that particular segment. Suppose if it's a case of a prostate pathology, might have the opinion of a, if we say this guideline has been curated by Dr. Mahul Amin of XYZ, who wrote the TNM classification, trust automatically increases. And here to start with, we are talking about common cancers. We're not talking about the extremely complex cases. That's one. Second, in diagnostics itself, there is also an unbundling process. Like say, while the wet labs are distributed, the informatics is centralized. So you have all of it in a GCP cloud. The people can sit anywhere. There is a maker, checker, validator type of process. The assays are whatever is built or not built. Go through a regulatory process if you're getting to a CVID, IVD, and or a companion diagnostic process. There are very well laid out guidelines, so to speak. And even for some of the elements of a technology correction and or a correlation, every state or a country has its own versions of it. Suppose if you want to go to the US, you have to comply with an FDA Part 21 and a high trust SOC 2 type of regulation. So in that sense, as we have seen in most industries, the hardware is getting to be software and software to an operating system. We have seen that in automotives. We have seen that in currently electric vehicles in a big way. So in that sense, medical and medtech for us, by virtue of initiatives like this, offer a unique opportunity for us to hopefully leapfrog a generation in terms of how we approach this.
So this only covers the trust part. How will you ensure the ownership of the diagnosis if it goes through multiple steps?
That's the in our approach of engaging this, we all historically are proprietary, right? What is it in it for me, myself, now? When we take that I part of it for a moment and then say, first let's try to solve the problem. We'll figure out what the economics or economic incentives are at a later point of time. In terms of ownership, the way we have approached it is the following. Like say, what is your IP is your IP. What is my IP is my IP. Anything that we jointly develop, it's a joint IP. An academic researcher has the first right to be the first author. We are not seeking the first authorship even remotely. In that sense, first author and or corresponding author for him, friends, family, everybody, no problem at all. And somebody's laughing here. Any friends and family also joining papers, is it? I joined my friends' papers, so I acknowledge in all honesty. So what I'm saying was, when you separate those two out and have a notion of a shared value co-creation, magic happens. Because at the end of the day, when you're starting with, you're seeking nothing. And many of these institutions as well, when you say what is in it for them or what is they also realize that they can't solve it on their own. None of us can solve it on their own. So by design, you would have to collaborate with somebody somewhere who probably has more knowledge and or awareness than both of us combined. And we'll get there. It's an experiment. We don't have all the answers. Our own commercial agreements vis-a-vis what we were before, vis-a-vis now, are very different from what we started 3 years ago. And our board said, so long as you don't lose money, it's fine. Don't lose your shirt and come here. So.
Hello, sir. Very interesting talk. Thank you so much. So I have the question regarding the challenge you pointed out, especially regarding the community delivery. So in India, people have this sort of mindset that, for example, if you ask me to have like a risk assessment test of cancer, I may sign up for that because cancer is so deadly in our minds. But it's not the same for the other diseases and all. So I want to have your opinion or your comment on, because these kind of challenges apply to other problems also. So where do you see that how as a sector, healthcare sector or system, how are we moving forward to tackle this?
See, it took 8 months to convince my wife to take a cancer screening test. Of course, one can say convincing your wife is the most toughest part of anything in life. That's a separate issue. But having said that, it's a work in process and a journey. Once we start breaking the myths of the degree or the association or the familiarity, let's take a simple example on screening. Like say, every woman above the age of 25, if she has not been vaccinated in her childhood, should undertake a HPV DNA testing. It's not that as if it is positive you have cervical cancer or anything. But more than the positivity, the negative predictive value of it is huge. You can assure her that next 10 years you're fine, because you're doing on the back of a molecular characterization of diseases, not a morphological expression. As science has told us, morphology comes many later when you see a radiographic evidence of disease. Molecular tells you much in advance, and that too when you do molecular, and that too when you get to a NGS/whole genome sequencing, you know the risk profile of it. Even in cancer, the biggest challenge with early detection cancer tools is, as Dr. Burdway of TMH tells, not every tumor might lead to mortality. And there are a lot of people who have stage 3, stage 4, but gone on to live their life as if nothing happened. You would need to look at whole range of other information that exists, which is often not easy to detect and or predict. That's one. Second, many of us from our own behavioral side, we assume we are going to live forever, right? And if any disease comes, it's not going to affect me, it's going to affect my neighbor, uncle, aunt, but not me. I'm fit and fine. Whereas for all our machines, every 3 months we want to, what you call, change the oil of our car, engine, or X or Y. Whereas the human body is the most powerful engine. We often abuse or disabuse it, because many things like willpower is more powerful than willpower. We can't overcome it, right? We find it quite tough to do it. But at the same time, if there are certain characteristics or characterizations that one lives with or gone through in their early stages of life or continue to do in their life, these things will come up, period. When it will come up, how it will affect you might be each individual's choice. But the fact that they will come up, science has periodically told, you will have it. That's it, period. So in that sense, for us, it's a lot more behavioral than medical. And in terms of a behavioral challenge, if we apply that to some of the other industries, in most cities, helmet was not a norm. I don't know what is it in Kanpur today, but in Bombay, without a helmet, you can't drive. Seatbelt was not a norm for many years. Now, after the Cyrus Mystery episode, seatbelt in the rear seat also in Maharashtra is mandatory. So it is changing. So regulation helps, policy helps, but more than that, personal compliance and ownership of it, there's no substitute to it even remotely. Thank you. Thank you.
Okay, so if there are no more questions, I would like to thank again Dr. Venkat for such a wonderful presentation. Thank you so much. Thank you.
First would be Pratyusha has sent over for the BTech students, right? Pratyusha. Yeah, Pratyusha.
So we have now the next session, which will be lightning talks by students, and the first speaker will be Tanay Visbash. Tanay, and he will talk on molecular landscape of genomic alterations in Indian prostate cancer patients. You have 5 minutes. So yes.
So good evening everyone. Welcome to this MFCEM inaugural day talk. So the title of my talk is Molecular Landscape of Genetic Alterations in Indian Prostate Cancer Patients. So to begin with, so this session is about molecular medicine. So what is molecular medicine? So molecular medicine uses different techniques such as physical, chemical and other bioinformatics or medical techniques to identify a cause of disease and comprehend the disease and help in better strategy. So this area medicine has shifted from one size fits it all to a more precision and precise approach. So precision medicine takes into account of different techniques such as multi-omic techniques, for example genomics, transcriptomics and other bioinformatics tools, to identify a cause of disease and stratify the disease according to patient's conditions. So what can we do to identify and use this precision medicine approach? So previously we have explored the molecular subtypes of prostate cancer such as TM-Precis2 ERK fusion, RAP gene fusion in Indian patients, and identified that there is a disparity with their molecular alterations compared to other published Caucasian and other cohorts. And interestingly, we found that RAF kinase gene fusion has a higher frequency in Indian patient compared to other cohort. So this suggests that there is a racial disparity within the prostate cancer subtypes. And likewise, TCGA, the Cancer Genome Atlas, has shown the molecular subtypes of prostate cancer, where they have identified that 74% of the tumors harbors one of these 7 subtypes of prostate cancer, and they have also identified 25% of these tumors harbor actionable alterations. So thus in-depth characterization of molecular landscape from Indian prostate cancer patients need to be explored, since this is completely unexplored. So to begin with, we started this analysis. We use whole exome sequencing of 32 Indian prostate cancer patients along with their matched blood samples to identify somatic variants and unknown molecular drivers. So we found that overall tumor mutation burden of our cohort to be 0.97, which is quite similar with the TCJ Pratt cohort. And we identified that most of these somatic mutations fell into this missense mutations category, and also a larger proportion of single nucleotide variants. Further, we classified these variants into one of the six classical subtypes, for example, and found that C2T transition event to be more profound. And this oncoplot shows the top 20 frequently mutated genes of our cohort, and interestingly we found RYR2, TP53 and SPTA1 to be most frequent. And we also found several genes that have already reported in other cancers in our cohort as well. And interestingly, we found that mutations in many actionable genes such as CDK12 or APC or ATM genes, which are found the patients that show this kind of mutations that can be used with certain drugs such as olaparib. And also we found out many mutations that have not been explored in other cohort and not reported in other DBSNP or cosmic databases. So this shows the uniqueness of this genome. And later we also compared these mutation frequencies with other published TCJ and SU2C cohort, and found out that there are several genes that shows this frequency is much higher or lesser in our cohort. And also we found out many mutations, for example in BRAF1 or CANSL2 to be uniquely mutated in our cohort, which suggests that the presence of novel somatic variants in prostate cancer of Indian genome. So thus we can conclude that the use of oncogenomic approaches would help us in stratifying the Indian prostate cancer patients and to develop better therapeutic strategy based on each single patient mutational profiles. And as a future prospect, the integration of this whole exome sequencing data with transcriptome and other methylation profiling data would help us in designing better therapeutic approaches, and also we may identify some unknown molecular drivers of prostate cancer. And further validation of these particular mutations or these genes and their functional characterization should be done, and that will help us in the development of better diagnostic and treatment strategies. So I would like to thank my mentor, Professor Bushra Atik, and my present and past lab members, all the BSB staffs and faculty members, and my friends and family, and most importantly, the clinical collaborators with whom we could not have done this project, and the funding agencies to support our work. So thank you everyone.
Thank you, Tanay.
Yeah.
The next speaker is Simran Singh.
Hello everyone, I'm really excited to present my recent findings, that is on single-cell transcriptomics and epigenomics atlas of pancreatic islet during different stages of insulin resistance. So to the never-ending list of global crises, obesity has managed to secure its position in this list. And to our surprise, obesity can cause a global economy as equal as a war or terrorism. So prolonged obesity has multiple root causes, and apart from that, the prolonged prevalence of obesity can lead to multiple health conditions as well. One of them is type 2 diabetes. So when we hear the term type 2 diabetes, two things come to our mind, that is the insulin or pancreas. And if the body is not producing enough insulin, the body will end up having type 2 diabetes. So talking about pancreas, the whole pancreas is not involved in the production of insulin. There are certain group of cells called islet, pancreatic islet, which are actually involved in the insulin production and rest other hormones. So, however, in literature, the journey from obesity and the involvement of pancreatic islet to type 2 diabetes is not very well explored. There are multiple multiple milestones there that are yet to be discovered. So to understand all those molecular events that is happening between obesity and type 2 diabetes, we designed a very interesting experiment where we actually worked on two groups of mice. That was one kept on regular chow and another was kept on high-fat diet. So this prolonged high-fat diet intervention can lead to insulin resistant and ultimately can lead to type 2 diabetes. To actually understand the correlationship between this pancreatic islet and human islet, we actually use the publicly available pancreatic islet data from control, obese and type 2 diabetic individual. So overall idea of the project was to look into the transcriptomics and epigenomics at single cell level. And interestingly, in our data set, we found that the pancreatic islet from mice encloses a wide variety of cell type, and we actually characterize these pancreatic cell types based on canonical marker, that was endocrine and non-endocrine. But the most interesting part was that when we compare our data set with human pancreatic islet, we found that the cell types were highly conserved. Now here comes the another very interesting thing about our data set, that was the emergence of different population at different dietary intervention. As you can see, there are few population which emerges very differently upon high-fat diet and regular chow. We also see differences within the high-fat diet intervention at different time point. And however, this diversity was not only limited to the cell population, we also saw that at the different dietary intervention, the cellular interaction between these cells were very different at regular chow and high-fat diet. Now, our interest shifted towards the endocrine population, that is alpha, beta, delta, gamma. And here we found that the alpha cells were of great interest. These alpha cells were highly diverse in nature, and as we know, alpha cells are basically involved in the production of glucagon, but in our data set, it was something different to say. So here we found that the one population was the canonical one that was actually involved in the glucagon production. However, if we see at the second population, that was mostly polyhormone in nature. That means it was producing many hormones other than the glucagon. So even in our trajectory analysis, we found that the second population was trans-differentiating in nature. That means it was actually emerging from different alpha cell, that is the canonical alpha cell, to become different population of alpha cell. And here we see different transcription factors which actually give rise to this trans-differentiation. Now, another very important cell type was beta cell, as beta cell is involved in insulin production, so it was of great interest. So we interestingly found three different beta cell type, out of which the third cell type was of great interest. Here we found that the third population was actually involved in glucose homeostasis, inflammatory responses, and carbohydrate stimulus. But the most interesting thing about this population was the plasticity. This population was highly plastic in nature. If we see at the plasticity between these cell type, we will find that during the high-fat diet intervention, the cell behaved in a very different manner as compared to the RC, and the plasticity was only introduced in high-fat diet condition. Overall, the results suggest that the pancreatic islet at different dietary intervention behave differently, and most importantly, they are very different at the prolonged period of high-fat diet intervention. Overall, I would like to thank my PhD mentor, Dr. Sai Prasad Paidi, and other collaborator for providing me this great opportunity to work on this project. And last but not the least, I would also like to thank the funding agency for supporting our project. Thank you so much.
Thank you, Simran, for such a wonderful talk. So the next speaker is Sattumi Chakravarti.
Good evening, everyone. My title for today's talk is NF-kappa-B PJNK inhibition mediated regulation of cartilage homeostasis by a combination of doxycycline and IGF-1 that synergistically attenuates osteoarthritis. So osteoarthritis is a debilitating joint disorder which has affected 7% of the global population, and it causes severe pain and immobility in the joint due to the gradual erosion of the cartilage lining the bones in the joint, and it is ranked as the 11th highest contributor to the global disability. The current treatment options based on the severity includes awareness, physical exercise, and administration of drugs which generally treats the symptom, and late stage includes surgery which may give rise to inferior cartilage, and TKR, which is total knee replacement, which is associated with secondary complications. However, out of the non-radical or non-surgical approaches, these do not treat the underlying disease condition, and therefore there is a requirement of agents that can halt or reverse the disease progression, and such agents are known as disease-modifying osteoarthritis drugs. Unfortunately, there are no licensed DMOADs. So to find out DMOADs, it is important to understand the pathophysiology of the disease. I'll be giving a brief overview, and it has been known from the literature that triggers such as trauma, etc., it disrupts the joint homeostasis, and that causes certain changes such as synovial inflammation, cartilage degradation, and subchondral bone remodeling. In the healthy condition, there is a balance which is maintained between the anabolism and the catabolism. However, in the osteoarthritis condition, the balance shifts towards catabolism, and there is secretion of matrix proteases such as aggrecanase and MMPase. So we hypothesize that by altering the joint homeostasis through a combination of pro-anabolic and anti-catabolic agent can be a disease-modifying treatment strategy. So to screen certain agents, we first developed a goat ex vivo OA model and screened few agents, and out of which we selected anti-catabolic factor doxycycline and pro-anabolic factor insulin-like growth factor. And then these agents in combination were treated in cartilage explant which was taken out from goat knee, and osteoarthritis was induced with IL-1 beta, and the treatment regimen was provided for 14 days. We performed histology staining and observed that there was higher presence of sulfated glycosaminoglycan, which is a matrix marker, in the combination group. However, and when we performed SGAG release assay, we observed that it was significantly reduced in the combination group. Similarly, we also performed immunostaining for matrix protease and observed that the expression of MMP was significantly reduced in the combination group. Then we tried to understand the mechanism of action of the combination. So we studied NF-kappa B, which is a catabolic pathway which has significant role in the progression of osteoarthritis. Here we observed that also NF-kappa B was significantly reduced in the combination group. So then we tried to understand if the combination mediates its effect through NF-kappa B only. So we selectively inhibited NF-kappa B. And then we observed that in NF-kappa B inhibited group, the SGAG release was more in comparison to the combination group. And similarly, we performed immunostaining for ADMTS and MMP, and similar effect was observed. So from this, we concluded that anti-osteoarthritic effect of the combination may be partially mediated through NF-kappa B and possibly through some other pathway. So then we studied another pathway upregulated in osteoarthritis, which is the JNK pathway, and we observed that JNK was highly activated in the IL-1 beta group, which is observed by the yellow color, and it was significantly reduced in the combination group, which is observed by reduction in the yellow color. So from this we observed that we conclude that the restoration of the joint homeostasis is mediated through NF-kappa B and PJNK pathways in goat OA explants. Then we corroborated this result in human OA samples, and here we observed that out of 4 human donors, 3 of them showed reduction in SGAG release in the combination group. Similarly, we also performed immunostaining for matrix proteases and observed similar results. So to conclude, the treatment with pro-anabolic and anti-catabolic factors shows promise as a strategy for amelioration of OA in clinically relevant models. The future prospect includes evaluation of the translation potential of the combination in suitable animal models. Finally, I would like to thank IIT Kanpur and MFCEM for the funding, and Ministry of Education for my fellowship, and all the TEDdy Lab members, and finally my guide, Professor Katti. Thank you so much.
Thank you, Saptami. So the last talk will be given by our BTech student, Pratyusha Chakravarti. Pratyusha.
Good afternoon, everybody. I'm Pratyusha, and I'm a 4th year BTech student in BSB department, and I would like to share the work that I did with Professor Rong Lee at MBI at National University of Singapore during my summer internship. The topic of my work is how different stresses induce protein aggregates of different subcellular localization. So we know protein undergoes folding from a primary structure to a functional structure, but in this process, misfolded proteins are also created, and these are removed from the cell via multiple protein homeostasis mechanisms. But when there is stress condition or during aging, these misfolded proteins are often not removed, and so they aggregate together to form protein aggregates in the cell. So, but protein aggregation has recently been viewed as a protective mechanism against disrupted protein homeostasis, which means that if these aggregates are localized to only specific locations of the cell, then these can be removed conveniently and would not hamper the rest of the cell. So we can also say that aggregate formation is probably a regulated process that requires specific amount of energy input and also occurs at only specific cellular locations. So in my project, we wanted to see if under 2 different stress conditions, one is by heat stress and one is by proteasome inhibitor Mg132, there was any difference in the type of aggregates formed or in the location of where the aggregates were formed in the cell. So we used the RPE1 cell line and we transfected them with the aggregate marker and the mitochondria marker, and we seeded them, and the two kinds of stresses were applied, and then they were imaged through confocal microscopy. So what we observed was, under MG132 condition, the aggregates were very perinuclear and localized around the nucleus, whereas under heat stress, these aggregates were scattered all across the cell. And we see the images where they were after 6 hours of stress and after 12 hours of stress. And we also plotted the distance between aggregates and the nucleus and aggregates and the mitochondria, and we see that both are lesser in MG132 condition than under heat stress condition. So this was the work that I did, but this opened up a lot of opportunity for future work. One was to find out the cellular mechanism that was responsible for this perinuclear localization of aggregates under MG132 condition. And then the lab that I worked with also established earlier a mechanism by which aggregates would adhere to mitochondria and then could be removed by being uptaken into the mitochondria and then degraded by mitochondrial enzymes. So they termed this mechanism as magic, and they proved that it could exist in humans. So now that we see that aggregates and the mitochondria have very less distance between them under the MG132 condition, it is possible that this could facilitate magic. Another thing that we did was we disrupted actin contraction using the Y27632 inhibitor, but we didn't see much difference in the perinuclear morphology. So we can say that actin flow is probably not involved in this mechanism, but this obviously is up for future work. So I would like to thank my mentor Wu Hongi and T. Han for their guidance and support, and my PI Professor Rongli for welcoming me to her lab, and the MBI intern program and my other interns who helped me throughout the stay there. And thank you so much.
Thank you, Pratisha. So with this, I close the first session that is on molecular medicine, and I would like to again thank Dr. Bao, Dr. Venkat, Tanay, Simran, Saptami, and Pratisha for such a wonderful presentations. We will break for tea now, and we will again gather back for the poster session. I think it's outside. So thank you very much for coming.